Yeah, it's the only thing that seems to work. So.
Hey Bradley, what's
Hello,
up?
how are you? How are you?
I'm fine. It's a long time since
Yeah.
our past meeting.
Oh yeah, how are you?
Yeah, I'm fine. Just busy with some of the tasks. Like I got some internship, just doing that.
Okay. Good.
Yeah, and currently working on the digital bachelor's project.
Okay.
Yeah. And can you tell me what is the specific task of the project? Like, what is our primary motive to doing this project? I asked Ujjwal about that. Like, he told me that to detecting several diatom, like specific movement and all other pattern in that project. So, is that the motive?
Yeah, I'll talk about it a little bit later, in more detail.
Okay.
But yeah, we'll talk about it. But yeah, basically that's the idea. Yeah, so nice to see you again. Susan, hello.
Hi, I bought a rather expensive microphone. See this? That they all seem to be about the same price. This is called Blue.
Okay.
So now I have to answer the phone. So
Alright.
I'll go.
Alright. Hello, Mainak and Tharun and Aayush. How are you guys doing?
Yeah, I'm doing really good.
Good.
Hi.
Hi. And Aayush. And I've been communicating with a number of you on Slack about your proposals and other things, and I'm getting to them. I'm working on it as we go along, and so I'm just giving comments in documents and answering messages. So you'll be getting those soon. So welcome to the meeting. I don't know how many. People will be coming in, I guess, over the next maybe 10 minutes or so. Usually that's how it works. And we'll just get right into some of the materials here. Hello, Tom.
Hey.
How are you?
Good. How are you doing?
All right. It said Tom P, so I was a little disoriented, but I know what it is.
Yeah, I don't have a camera on this machine. I tried one with a camera, but it didn't work.
Oh.
Yeah. Maybe I'll try and switch over.
Okay, well, yeah. Welcome.
Well, yeah, good to see everybody again.
Yeah.
Yeah.
And Jesse's here. Hello, Jesse.
Good morning.
I don't know. Well, Shruti's not here right now, but like Tom he's worked with the group over a number of years, and he's one of the people who's done a lot of work on cellular automata and the morphozoic platform. So we've talked a little bit about that in recent meetings. So he's doing a lot of stuff with artificial intelligence, with like biomimetics. He gave a talk in the group last year, so some of you are probably hadn't joined yet, and so he's interested in that area. So if you want to, he'll be in the Slack channel for open or for DevoWorm. If you want to ping him, if you're interested in like the cellular automata area or some artificial intelligence topics. Mainak says hello. So welcome to the meeting. We have a number of things today. I wanted to point out, first of all, let me share my screen. We have Akshay. Hello, Akshay.
Hello, Bradley.
Hello. So we have a new publication out. It was yesterday, I think it went live. And this is a periodicity in the embryo paper. So this is Ujjwal, myself, and Jesse were the authors on this. And Susan didn't want authorship, but she's in the acknowledgments. And this is finally on biosystems. This is the special issue on waves in development. And so this is the final sort of stub for the publication. It goes through a series of stages here. The first stage was as a preprint, and we had that out for a while, and then we revised the preprint, and then we did the final version, which is in Biosystems, and there was like a in-press version of that, and now it's the final version. So it's a pretty good paper. It's about the timing of cell division and cell differentiation in two different embryos, C. elegans, which is the nematode, and Indani auraria, which is a zebrafish. And those are two model organisms, but they're very different modes of development and different things going on in their development. And so, we can look at those two models. And then we can also simulate cell division using a simulating data set that we created for this purpose, and looking at sort of the speed of division and that sort of thing. So you'll have to read the paper to get the details on that. But it's I hope people enjoy this paper. It was something that we were going to do maybe 2 years ago, and I got put on the shelf. And then finally there was an opportunity to go forward with it, so we took the opportunity, and now it's out. Okay, need to get access. Yeah, let's see. If you go to the DevoWorm Weebly site, thanks, Tom. If you go to the DevoWorm Weebly site, I actually have the Biosystems version, which is pretty similar to it, but it's, you'll get a sense of what's going on. Hello, Bikram.
Yeah.
So that's periodicity in the embryo. I can post a version of it in the Slack channel as well. Next thing I'd like to talk about, actually, this is something I pulled up. I had this in the queue of things that maybe need to be done. I'll talk about that in a little bit. This is a paper called Morphogenetic Patterns in the Theory of Deep Learning. And I have a couple people on here as authors right now, but we can add more authors if people want to contribute. So the idea of this is it's about sort of deep learning and how deep learning networks can tell us something about development, specifically the creation of morphogenetic patterns and maybe some other phenomena. So we talk about some of the aspects of deep learning in GANs, and then kind of going through maybe how some of those things might provide insights into development. It's pretty rough. It's a abstract still with some references, but we'll flesh this out over time. I just wanted to bring this up because I want to give people a chance maybe to get involved with it and see what we're doing under the hood. This is something that I've shared with a couple people in the Slack channel, and it's been a while since we revisited it, so there isn't much here right now, but I'm trying to get this together so we maybe can submit it to a conference or somewhere else. So we could probably submit it to a conference as the abstract version, but we might also want to do a full paper on it. Okay, so next thing I'll do is talk about the onboarding guide. So for those of you who are looking to join the group as a member of the, maybe as like a GSoC person, or doing a proposal in GSoC, or looking to get involved in the group, we have this onboarding guide that's now available. And this is, let's see, Mayukh contributed to this, and Krishna contributed to this, and I pulled a lot of stuff together here, and there's a lot of stuff that we've had in different places. And so the purpose of this document is to give people an idea of what's going on in DevoWorm. So this is, I have to reload it. Okay, so the purpose of this document is to give people sort of an essential guide to DevoWorm, and the different parts of the project and what's going on, but with an eye towards like GSoC and contributing. So this is sort of a little bit on the OpenWorm Foundation. We have some links here. If you're not in the Slack, here's a way to get into the Slack. Here's the OpenWorm GitHub, which is different than the DevoWorm GitHub. This is the OpenWorm website URL and so forth. And then we have some resources for some of the OpenWorm projects. So, DevoWorm is one OpenWorm project. There are other projects like C302 and some other projects that are, doing a lot of different things with the worm, looking at, simulating the activity of neurons, looking at the connectome, looking at some of the other things going on, movement behaviors. And so those can be found here. I need to add a couple more resources here, but, and this is a talk that I gave at Artificial Life 2020 on OpenWorm. And so you'll find those things very informative. Then we get into the DevoWorm group and some of our things. We have a group website. We have our own GitHub repository. We have DevoLearn, which is an organization devoted more to like machine learning and deep learning, and analyzing like developmental data. And so we have all these things here. And then we have some introductory slides from Krishna on, how to maybe contribute to open source, how we participate in GSoC 2021. So we participate through INCF, OpenWorm. We belong to OpenWorm, but we participate in Google Summer Code through INCF. And so that hopefully that clarifies things for people, gives you some ideas about what is open source. This might be a little bit too, low level for some people contributing or applying to GSoC, but I think as a general resource, it's a good way for people to kind of, understand. Some people who come to the group don't really know what, open source contribution is. So we want to make that clear to people. And then here are ways maybe you can contribute to DevoLearn. This is just a process of how to put a contribution into play, going through, looking at what's going on, trying to find an issue to address. Maybe you create an issue, maybe you get in contact with people, but you then work on the issue. And we had a couple issues this week where people worked on them and submitted pull requests. And so now their pull requests are in, and so now you're formal contributors to the project. Then there are the project descriptions. So we had a question at the beginning of the meeting about digital basilaria. I know 3.1 is popular. This is upgrading DevoLearn. This is the DevoLearn software that Mayukh is maintaining. And this is his GSoC project from last year. This project revolves around working on that software platform, which is essentially a pre-trained model for analyzing developmental data. So, analyzing data for embryos, the C. elegans embryo, but other embryos as well. And, developing techniques for analyzing those data. So, this project, though, is upgrading the capabilities of that package. And so I think a lot of you are already aware of maybe some of the things that entails. There's a formal project description for all these projects on Neurostars. So I think a lot of you are familiar with that. But Digital Basilaria actually is, this is a project that started where we had this diatom. It's a single cell colony, so it's a bunch of cells that are behaving together and interacting. It's called basilaria. It's a group of diatoms that are these long rod-shaped diatoms that exist in colonies, and they move around in different ways in the water column because diatoms are marine microorganisms. And so we have a lot of video of these organisms, and diatoms are actually fairly common in aquatic environments. So, if you anywhere in the world you can find them, you can isolate them, and with some luck you can even culture them in a dish. And what we're interested in here is looking at these microorganisms, getting microscopy images, which we have a library of. And in this project, your job would be to analyze these images. So you'd be doing some segmentation, you'd be doing some annotation, and then working with the video. And then we'd be doing some things down the pipeline, which you might also be involved in, depending on your skill set, such as analyzing the data and, maybe building simulations or visualizations of the data. And so that's what that project is. The diatom world is, they don't have a lot of sort of digital models of organisms here. We have in DevoWorm, the reason it's called DevoWorm is because we focused on C. elegans, which is a very well-characterized model organism. So C. elegans, there's a lot of secondary data floating around. There's a lot of data about different aspects of C. elegans. Surprisingly little about C. elegans in the wild, but by comparison, diatoms are pretty poorly characterized in that way. So our goal here is to sort of build a digital diatom. So it's an organism that's not a model organism necessarily, where people study it to study disease or other things. People study diatoms for like biofuels and things like that, but there's no digital characterization of it. So, and I have in this onboarding guide, I have a couple of links. This project has been worked on previously by Ojwal Singh, and he's developed this platform called DevoWorm AI, which actually has some of the models for digital bacillary, but also for some other models, machine learning type models for analyzing, microscopy images and things like that. There's also a link to the GitHub repository for digital bacillary, which is, not in the newest state possible, but we have some references there. So it hasn't been updated in a while is what I'm saying. There's some resources, though, here for you if you're interested in working on this and maybe doing an application or working with the data. We have some raw video data, which is microscopy data. So we have microscopy that's in the form of video files. So you'd have to take the video files apart and analyze the individual images. Or, if you want to recognize things through video analysis, there are different approaches you can use to segment the images. And then we also have temp delimited versions of the data. So these are data that were extracted in our previous years of work, and those are available for people to look at and to maybe use to validate their results or whatever. We have a paper here which is on the bioarchive, and that talks about a lot of the analysis that's already been done. So it gives you a sense of what we're trying to do, at least to some extent, at least in terms of the image segmentation. And then there's this recent presentation on bacillary movement. This is the presentation that we gave to Neuromatch, which is a conference, an online conference. And this had to do with looking at bacillary behavior. And so this is a model of behavior for bacillary. And so you can find all those links here. Finally, there's this digital microsphere project, which is it's a project where we use a special type of microscope where we capture an embryo from like many different sides at once. So we're observing it from many different projection angles or points of view. And then we take those points of view and we want to stitch them together into an atlas or some sort of like visualization that we can explore. And so there are two papers here on this. One is on the what we call ball microscopy, and Susan is actually the person who's heading up this effort to acquire this embryo data through using this ball microscope. So she puts an embryo in the stage, and then she has all these microscope, these cameras around different sides of the embryo, and she's able to get a bunch of different angles at once. And then the idea is to stitch these images together into an atlas of some type. And this paper describes this vision in terms of sort of like Google Maps, but Google Embryo. And this is about 10 or 12 years old, but I think you'll get the idea of what we're trying to do. There's a notion of like creating like a map or something you can explore, a surface you can explore. And there are different ways you can do this. So it isn't just, creating something that looks like Google Maps. It could be any kind of like, you could take like a sphere and something that you could move around and turn with a cursor. It could be a flat map. It could be some other type of visualization. But the idea would be to take these data and project them to something that's explorable. And so there are a lot of things that this can lead to, a lot of different insights to be made, a lot of different ways to display the data. So there's a lot of potential here. So that's almost all I wanted to talk about with the onboarding guide, except that I'm also developing some of the biological resources here. So what we also want to have here at the end of this onboarding guide is I want to add some references in on diatoms, because we don't have that yet. And I think that would be useful to people. But we do actually have, and this is from years past, we do have some references to C. elegans biology. So if you're not up to speed on your C. elegans biology, you can put it in here. And there's a general FAQ, so you can find out more about like some of this language is like, I need to be more specific. These are C. elegans data sets, but I think we're coming along on this. I think this will be a good resource for people just joining the org. And if you're interested in contributing resources to this, please feel free to, make a pull request or contact me on Slack or, whatever, and we can keep improving this onboarding guide. So what do we have? A bunch of comments here. So after the onboarding guide link, let's see. Yes, can you please upload the recording of this meeting? Yes, that happens every week. Jewel of the Sea, Debo says. Susan says, how big are the basilaria? So they're about 90 microns long. Maybe a little bit more, maybe a little bit less, depending on the... Well, in basilaria, they're that long. It depends on the diatom. Diatoms have many different morphologies. Some of them are round. Some of them are these long rod-shaped things.
Like two categories, round and rod.
Yeah, there's several different categories actually, but those are the two main ones. So you have different genera and species that are they have different shapes. They're characterized by sort of silicate cell walls. So it's made of silica, which is different from a lot of other organisms. But they have this. They have some really interesting properties as organisms go. So they have these silicate cell walls, and they have their own cell division dynamics, and they have their own growth dynamics, and they don't. This isn't really developmental biology, but it kind of is, because these colonies grow and they move. So the Digital Basilaria project isn't really focused on the development so much, but in as much as they have a developmental stage. But I'll put some more information about diatoms up on the in the onboarding guide, so we can get some of that up there and make sure people are on the same page in terms of what's going on there.
So, final aim of the project is to create a digital version of the OpenWorm. Like the OpenWorm project, they created a cybernetic worm. So, for the digital basilaris project, are we going to create a simulation of the same kind of thing?
I think we'll probably have something similar. I think a better way to think of it, though, is like a digital characterization. So, we can actually take these organisms and we can image them, and then we can get some information about their morphology, and we put that into a digital format, meaning we can attach numbers to it. We can have like a model of its shape, its size, its like maybe location during movement. We have all these different parameters that we can store, and then we can reconstruct this maybe in a simulation. And that's kind of what we're doing with OpenWorm, with C. elegans, is we're trying to characterize all the neurons and the cells and the morphology of the worm. And then we're trying to get, a better characterization of the worm and its behavior and all this other stuff. And people talk about, making a sort of, building a digital organism. Well, digital organism is a lot of work. And of course, you're not going to get like down to the level of like genes or, particle physics, but you're going to get this model that is somewhat like approximating what you would see in nature.
Like a approximate digital organism or something, like we are taking a bottom up approach, that we are getting all the data from the real, like by this deep learning and machine learning stuff, and after that we are going to reconstruct this in a digital form.
Yeah. And that would be the ultimate goal. And of course, it's a long-term goal. It's not going to be done in 1 year. But that's
Yeah, like I think there is a lack of data. We need some more data regarding this. So, can you think like how can we collect? Like if there was be 100, I think there was around 100 images for the diatoms which are labeled.
Yeah. I mean, we're doing a very limited scope here, and we're actually getting the images from a collaborator. So, I mean, there are different ways and there are data sets online for, not for diatoms so much, but for other model organisms. And we have something called DevoZoo, which is a sort of a repository of different images that exist. So people will image, do high resolution imaging of different organisms, and they'll, make the data public. And so we have a repository of those resources and some other resources that we've kind of put in one place, and then we've processed those and created numeric descriptions of those things. And so those are all available as well. And I don't know. Yeah, I need to make sure that the DevoZoo stuff is clearly demarcated in the onboarding guide. But I think those are all good questions.
Yeah.
Yeah.
And I think it's a very good aim to that, like understanding the simple organism first, and after that going to more difficult one.
Yeah.
And I see your digital microsphere project as well. I see some of the recordings on the YouTube, and I think it's a really great project that we are going to try to create a map of the embryo morphology, and try to understand it by this, constructing the what you said, that Google Earth. It's the same kind of thing, like a map, what I understand.
Yeah. So the idea would be to take the data and make like a visualization that you can, because the idea is that usually in a study, if you took like an embryo and you put it on a slide and you took microscopy images, you would get like slices of the specimen, but it would be at one angle. So it would be at one orientation. It would be like sort of the top of it, and then
like 360-degree view.
Yeah, this would give you like a view, but also like over time. So in an embryo, things are always changing. And so the idea is that you can image the thing as it's sort of changing over time. If you have different samples that are different stages of development, you can get that information over time. And you can get different orientations of it at certain time stages, and you can make connections between different stages of time. So, if it's like an early stage embryo versus a later stage embryo, not only what's going on in terms of the divisions, but like, is it turning, if there's something going on like rotationally, you can capture that, or cells moving around at the top or the bottom of the structure, because they often do this where they move around the embryo during development. So all those things you want to be able to capture as well. There's the DevoWorm YouTube account. We have length 70 to 200 microns, width 5 to 8 microns. That's for the basilaria size.
And I think it's a good resource for the diatom.
Yeah, it's a good resource for diatoms. So we've got some people interested in proposing projects on that. And so Susan had a couple things to say about the ball microscope. The ball microscope has a 1 millimeter field of view, so just wondering if I could get 3D images. So these are, you can also make, three-dimensional images as well. Depending on how you can play with the images. We can get a lot of information out of these images. The ball microscope can be adjusted so that it has several centimeter field of view as well. So we can get very wide angle shots in the ball microscope. I don't have a picture of it, but I've shown it in previous meetings. It's basically this.
There is an image. I see that. It's. I think some of your members sent the image of the ball microscope, and it's like a ball and like a football. If we can imagine that is a football, and into the microscope, if we put like a sample of the embryo, and we can look at that, like there was some pen kind of like a pen or pencil kind of structure around it, and via it the information is going to the computer or something like that.
The pencil structures on it are actually the microscope cameras.
Okay.
So, and I've got, how many? I can go up to 14 microscopes with that structure, but right now I only have 9. Okay. So, just 9.
Yeah, that's great. And then Akshay asked, yes, Susan, I was actually planning to do sort of a Mercator projection approach, which we may stitch all the embryo images together. So yes, an example would be like a map of the world where you have latitude and longitude, and then the coordinates would be like a 3D projection. Yeah, so that's a good example, is like a map where you take like the surface of the sphere, which is the Earth's surface, and you pull it off and flatten it out. And so you need to do, you can do that, but you also need to have a coordinate projection, because when you flatten it out, you're going to have a lot of distortions. And so there actually, aside from the Mercator projection, there are other types of projections that people might be able to do, and there are projections that are better in terms of capturing the information at the top and the bottom, at the poles of the image. And so, like on a regular flat map of the Earth, for example, in some projections you got a lot of distortion at the poles. So things appear to be much bigger than they are, and things in the equatorial regions appear to be much smaller, which is a problem if you want to know what's going on at the top of the image. And there's a lot at the poles in development, so it's it's important to get that information. But there are other projections that you can use. But I think using those kind of 2D coordinate projections as a way to approach and analyzing the data. There are other ways as well, like being able to. There are a lot of mathematical models that one can use for doing different things, and we can talk about those if you're interested. But I don't have any good references right now, but those exist. And so, let's see, Akshay also asked, otherwise an alternate approach would be to segment the images using OpenCV to obtain the embryo part only. Later on, or can be used to obtain each cell of the embryo. After that, the stitching can be done of the obtained cells to make a 3D plot. So I've put some data. I don't know if it's in the onboarding guide. It should be in the onboarding guide. I've made a link to some of the data. There's some data in here, I believe. Maybe I don't have it in this one. Well, there are some data that I've made available, and I will put the link in the onboarding guide. And this is like a sample of data that Susan gave me, and it's just like a sequence of events. So like you have this sphere, and it's not like every angle. It's just like a single. It's an earlier version of this where there's like a flipping. She has what she calls a flipping microscope, which is where basically the embryo rotates in water, it's turned upside down and then it turns upright. And so by doing that, you can see most of the surface of the embryo, but it rotates over time. So you have to map these images to some sort of rotational movement. And so, someone kicked out Debo. I don't know what happened, but
maybe can you turn off the anonymous mode or something like that? I think it will everyone can have the moderation control. So yeah, like Google Meet, you can only take control.
I'm not sure why it's doing that, but yeah, I don't know why I did that. I don't think there's anything I have on my end that I can.
Yeah, but okay, no.
Okay, yeah. All right, sorry. So yeah, Susan, can I have your email of that? Do we have any presentations today? We may have some presentations. So yeah, does anyone have anything to present today, or? I don't know if anyone doing any GSoC stuff has any presentations they want to do. Last week we had a couple.
I'll present that, Bradley. Last week I couldn't present due to the change in the timing of the meeting. So I was supposed to present the meeting.
Okay, go ahead.
Can you see the screen?
Yes.
Yeah, okay. I worked on the 3.2 project 3.2, which is the basic area segmentation. So basically, what I did was, firstly I curated a data set and generated the annotations using various methods such as binary thresholding, canny edge detectors, and manual labeling. So as you can see, the individual cells on the right, which are segmented lightly in white background, and then I went ahead and trained a segment unit-based deep learning architecture. So basically, in this architecture, I replaced the normal convolution blocks with residual blocks to improve the long-term connections as well as to improve the learning of the model. As we go deeper and deeper into the framework, there might be lots of information with the use of normal convolution blocks. So I basically replaced them with residual and identity blocks, and then I had the results here. So if we give in the input image, we get the prediction of the segmentation as this, and we have the corresponding ground truth for that particular image. And then I went ahead and also developed the GUI platform, wherein if you closely look on the left top side, you have an option to upload a particular image which you want to analyze, and then when you click to analyze button, the GUI runs in the background and generates the segmentation mask and then displays the input image along with the prediction to the user or to the end user who is using the platform.
Okay, that's very good. So this is the GUI, and this is so we have the DevoWorm AI, but it's it's still a work in progress. So I could see this as being something we could put into the DevoWorm AI platform. So I'm not really sure.
Hello, am I audible?
Yeah, you're audible.
So hello, hi Aayush. So whatever you have done, first of all, these are the images that you are showing as prediction in the GUI is from unit architecture.
Yeah. Like I created a prototype unit
Okay.
architecture and I just trained it
Can
on
you
say
can
100
you
epochs.
zoom it a bit? The prediction one, and you have used Streamlit, right? For app,
Yeah, for the GUI platform. Yeah,
you have used Streamlit. Okay.
yeah.
So can you zoom it a bit?
Mm,
Maybe. Yeah. Oh, if that was the same image. So yeah.
yeah,
So
they
like.
both have the same image. It's a bit
So
different. Yeah, you can say this one is more zoomed in.
yeah. So I wish like even though work is pretty good, but like the objective of our problem is just to clear. Is like we don't only need to like segment these using binary marks. We need to analyze them as well. Like we have to find parameters like the height of each cell, individual cell, how many cells you can identify, like how many. For example, if you are taking some sort of bounding box approach, how many boxes are there per frame, so that because like we have to take all these frames in continuation and then we have to analyze like what is the predicted movement for this particular norm that is being shown in the video frames. So like whatever you have done, this is a good starting, but you have to like evolve it something so that we can analyze it. Like you can't really analyze like each individual cell in the diagram. The segmentation is pretty good, but I think you are getting my point.
Yeah. Like I was just working on the prototype last week. That's why just to lay out the groundwork for this, so that I can build upon it later. So like on developing the GUI platform as well as the unit architecture, and now I have some groundwork to work on. I'll definitely start working on all the aspects which you mentioned.
Yes, sure. Like, I just want to make sure, like, you are clear with the problem. Like, if you are clear with the
Yeah.
problem, then it's a good one. No issues. Thank you.
Thank you, Ujwal. Yeah, that was good. Thank you for clarifying some of the aims here. So Ujwal worked on this in 2019, and he's done a lot of. He knows, kind of how this would proceed. Yeah, you're showing the masking. So I think it's masking, but also the annotation of the cells, individual cells, and being able to get those because the whole that's a whole colony of cells there, and so being able to get the individual because what will happen is the individual cells will move around relative to one another. So having the individual cells marked out and being able to annotate them, even because they don't really change shape, but they change position, and so. So good work. Anyone else want to present anything, or have any questions about this?
Am I audible?
Yes.
Yeah.
Okay, so Ayush, like, how large was the dataset in which you trained it?
Like I trained it for only one folder. Like I guess in the GitHub repo there were 5 folders. I guess containing the dataset. So I just generated annotations for the folder one, and then just to start the prototyping and all, I worked on that one.
Okay,
Like
that's fine.
around.
The thing is, like, how good are the IOU scores or the losses? Like, how good are the metrics for now?
Yeah, like I without any data augmentations, I was getting an IOU score of around 0.86, and after using few augmentation from the argumentation library, I was able
Yeah.
to get an IOU of around 0.929.
Okay, so which means basically a 92% accuracy, which is
Yeah,
pretty good, I guess. Like, we can like easily get 95 if we expand it to the whole data set, I
yeah,
guess. Okay.
yeah. And like if you use aggressive data augmentations as well as
Yeah.
model training architecture and tweaking around the models, so we can.
Yeah, maybe we want to use multiple models for like
Yeah, like an
99% ensembles. Yeah.
ensembling. So yeah.
Yeah, we could do that as well. You might try that as well, like, so that's
Okay,
just what
I'll
I
just
get from my
try.
side.
Oh, like initially, as I told you, just prototyping to lay out the groundwork about how things will proceed. So
Yeah, I
I,
get it. Like, this is just the boilerplate, right? You
yeah,
just,
right.
yeah, I get it. Great work.
Okay. Yeah. Thank you. So that's again, that's a good job, and we have a number of approaches that people are trying on these different data sets. So if you have anything to present, if you're doing work on putting your proposal together, or you have something that you're doing you want to show to the group, this is a good time to do it. If we don't have anyone else, I can move on to the next order of business. A comment in the chat here. Okay, pretty interesting. Yes, very good. Thank you. It's very interesting. So I want to talk next about this. We can talk a little bit about the submissions, but I did have this thing that I got contacted in by someone in OpenWorm who works on another project, but they're interested in putting together this thing they call the mathematics of OpenWorm, and so I'm trying to put together something called the mathematics of DevoWorm. And what this is it's like it's a way to sort of boil down a lot of the topics that we're doing in the group to sets of equations or models that people can sort of summarize and understand. So these are some of the ones that I have here, and we'll probably add more. I have a couple of different categories. I have developmental function, I have spatial complexity, and self-organization. Those are two categories. And then in the spatial complexity category, I have this model of an embryo where I have this set of parameters this is sort of the model that we do when we analyze embryo data. We have what they call cell tracking data, which is being able to track the cells and assign a specific three-dimensional coordinate position to each cell. And then you have things like time and division angle. So the time is when you capture the image in terms of how the embryo is unfolding. And then the division angle is when you have two cells that divide and start to move apart from one another. What is the angle of that? And so those are like 5 parameters that I've put together here. And we have a paper on this from 2014. So if you go to the DevoWorm website, you'll see a link to the paper. But we can have other parameters here as well. And so this is one sort of approach. Another thing that describes maybe spatial complexity is this idea of a cellular automata. So this is a von Neumann neighborhood, and this is the radius of the von Neumann neighborhood. So this is the equation for the radius of that neighborhood. So you can have von Neumann neighborhoods, which are the immediate cardinal direction neighbor cells. So it's like the cells to the right, to the front, to the back, and to the left. You can have many different orders of neighborhoods. So this is an order 1 neighborhood with a radius of, I think, 1. But you can have much larger von Neumann neighborhoods. So this is just a characterization of this idea, this concept of a nearest neighbor neighborhood. And what I'd like to do eventually is to take these, make sort of like figures that describe each of these categories, and then like write a paper or a short paper describing these things. And so, I don't want to just leave the visualization there. I want to actually describe what it means. And so this seems a little bit like out there now, but there's going to be some description of it later. Developmental function is another example, and for that we have 3 different sorts of data structures. We have complex networks, where in development we have node attachments. So these are like our embryo networks or networks of cells or something where we're adding nodes over developmental time. We also have neural networks, which of course we use in things like deep learning, but are also like in the connectome you see neural networks, which are configured a little bit differently, but they, that's the basic structure. And you have lineage trees. So you have this is a chart that describes cells when they divide and they form new cells or daughter cells, and then they divide again and they form daughter cells. And this recursive process is described by a lineage tree. So what I wanted to do with this is to sort of put together like the major equations and data structures involved in development, and then describe them. And the person who I talked to in OpenWorm said that, maybe we would submit this to something like Wormbase or Wormbook as sort of a document for the community of C. elegans researchers. But it would be more, general interest to people interested in like computational development and how to think computationally about development. So this is another thing that's sort of like we're starting out on it, and if people want to be involved, they can get involved. They can, contribute something to it. So then that brings us to the submissions list, which is the submissions that we have out and the submissions we might be making in the near future to different journals or to different conferences or to other venues. So, and those of you who've been in the meetings before kind of know the drill. There's this list of things. These are things that may happen or may not happen. We just need to kind of move them forward. So this is these green rows are things that are done. So we have a couple things that are already done. I started this list at the beginning of the calendar year, so we're already getting there. So we have a couple of things here. Evolution 2021, we've been talking about submitting some abstracts there. I think Krishna has one on Kill the Winners, which is something he presented on several weeks ago. And we also have this Euler Paths for Life, which is something I've been working on. I showed it in the meeting as well. If you're thinking about submitting to Virtual Evolution, this is the link to go to the submission portal. It should be open now, but the deadline is April 30th. So you'll want to work on your abstract and submit it by then. The cost is, it's not as much as it would be if it were in person, but you can check out the costs at the link at the meeting site. So again, we have, and people can submit if they feel like they want to have something they might want to submit to this conference. They can maybe put it on this list or go ahead and submit it yourself. But I'd actually like to revisit this Kill the Winners submission. If Krishna, I don't know if he's still here. Here he is. If Krishna wants to talk about that in the next week or two, maybe we'll get back and look at that. Actually, following up on the last one, Jesse says, I'm interested in how the math models work. Is it on GitHub anywhere? The answer is we don't have a GitHub repository on this, but this will be sort of like a next step. So stay tuned. Krishna, you were going to say something? You're muted.
Hi, hello everyone.
Hi.
Hi Bradley. Nice to see you. So yeah, I can present that thing, and plus we have another paper coming up that was on the bias. So I'd like to present it. Can I present it today?
Yeah, you can go ahead and present it if you want.
Okay. You can complete with the you can say deadlines, and I'll start.
Okay. Yeah. So let me go through this really quickly. So yeah, we also have the DevoWorm paper, which is something I've put on the back burner, unfortunately, but I'm going to work on it. And this describes our DevoWorm platform. So those of you contributing to Google Summer Code are pushing to the DevoLearn repository, and we have a bunch of new people who have issued pull or have fulfilled pull requests. I don't know if we have anything on the pull request list here, but this is the DevoLearn repository where a lot of you have been contributing. Thank you for that. And if there are pull requests open right now. Mayukh is taking care of all of those pull requests. Thank you for being a maintainer, Mayukh. It's hard, it's hard work, but it's good to be able to maintain these resources. And maybe next week we'll do a little bit more on the different pull requests on this. But suffice it to say, that's a main part of the DevoLearn platform, But we also have a lot of other things here. We have the general biological model, which is a machine learning model for analyzing different types of biological data. We have the C. elegans DevoLearn, which is it's a web app to support the C. elegans part of the DevoWorm library. So this is part of this DevoWorm AI initiative. And we have a repository of model organisms here, which needs to be worked on because we don't really have a lot of descriptors in here, but there's a lot of stuff that's coming up on this platform. So there's the software, but there's also the platform, and this paper is about this platform, and we're still working on it. But if you want to be involved, you can start by contributing to the DevoLearn software. And so we also have this growth form and theory of deep learning paper, which I showed earlier. We don't really have a target venue for that, but it's, it'll happen. We also have this bacillary non-neuronal cognition paper that is coming up. We have to write a paper by April 30th. I'm working on that paper. We put we submitted an abstract to a book chapter, which got accepted, and now we have to produce a paper. And that's where a lot of this bacillary analysis is going, into things like this. This is OpenWorm. Oh, there's an OpenWorm poster that someone else in the OpenWorm organization is interested in submitting to the International C. elegans Conference. I think that's something that we can just say DevoWorm wants to be involved with, but then maybe later down the line we'll actually put some content into that poster. And that poster, the International C. elegans Conference. The deadline for abstracts is March 25th, but the conference is in June. So we'll be talking about that in the next couple months. We also have this Boring Billion book, which is this idea about deep evolution and why things were static for like 1 billion years of Earth history. So that's still being worked on. I submitted a proposal for a talk on the DevoLearn platform to the INCF Neuroinformatics Assembly. So that's coming up in next month, I think. And we'll be talking more about that in the next coming weeks, preparing materials for that. And that'll hopefully be something that people can attend online or, however they decide to host it. And then there's this Mathematics of DevoWorm, which is this worm book thing, which is pretty rough right now, but we're getting it together. And then I wanted to point out for a lot of machine learning people, there's the deadline for NeurIPS, which is on May 19th for an abstract, and May 26th for a full paper. If you have, if you want to write a paper, time is pretty short on that, but you can submit something either to that or to one of the workshops, which have a little bit later deadlines. So that's the paper review. I just wanted to review those deadlines. And now, Krishna, would you like to present?
So, hello everyone. Can you see my screen?
Not yet.
Yeah.
There we go.
Okay, so here it is. This is a paper me, Bradley, and Chrissy are working on, and it's on bias in artificial intelligence. So I'll just give you a quick recap of what are the things that we are doing here. So in psychologists have considered that there are more than 200 bias in human brain. We tend to make a lot of assumptions, and we work on them without checking them and confirming them. So that's where a lot of stereotypes and a lot of bias comes into play. So you must be know about garbage in and garbage out. Same goes out with machine learning algorithms. If you are feeding an algorithm with wrong data, the output would be wrong. It won't be as efficient and as good as it should be. So the main 7 types of bias is, you can say, exclusion bias, sample bias, measurement bias. So there are nothing like, for example, if a person is designing an algorithm and he made a perception, he wants to see the things that he would like to see, then there is a quotation that you see the world as you want it to be. So here it comes into play. Then we have measurement bias, like whenever the data that is reported to us is incorrect, it is not accurate. Then we have recall bias. It's come in the labeling stage. And for example, like if you are labeling two things like gray, silver, and black, and for a silver color thing, we one time label it as black and other time label it as silver, then our data set is already having recall bias. So we have a lot of, you can say, 7 biases are here. So one of the major is this associated bias. It's generally applied in natural language processing, and a lot of gender stereotypes come into play because most of the data that is trained also contains the cultural stereotypes. Like we don't usually have in our training set women as engineers, and we are generally, for example, if you are feeding professions, so it was found that in Google translation, when men were linked to doctors, women were linked to nurses, and it should not be there because both men and women can be doctors. So that is something that should not occur. Like in some of the machine translation problems, when men were targeted as engineers, so women were not targeted as engineers. Instead, they were targeted as teachers. So there is nothing wrong being a teacher or engineer, but the translation that was happening was not fit for real world. And for example, that measurement bias, anyone who has looked on computer vision knows that we usually have a drastic drop of performance when our algorithm works on real world data, because the data set we are having is very good resolution data with good lightning and the sharpness and everything of that image is very good. But when you come into real world data, there is a lot of present noise. The backgrounds are not very much considerate, and so models the performance drops a bit. And this is, you can say summary of the paper that we are working on. And I want to present one more thing. It's not yet. Any questions from bias paper?
Well, I think it's a very good point that, like, even like a lot of the stuff we do in the group with respect to data is some of it's high resolution, but we also have to remember that we're dealing with very small samples relative to the actual biology that's happening. So I think everyone should keep that in mind as they're doing their work. But that's fine. Yeah, we can talk about more about that in meetings to come. So
yeah.
Just adding one point, like one of the main biases that I have encountered, like while working with the pre-existing model is all the models which are in the image generation are trained on a particular set of people. I am not very much aware how to say, but only the most of the people have white faces whenever they are making the models and all of this, and these models are performing very very poorly for the other people. So this is one of the greatest bad that the world is dealing with right now. All the models take can, fake generation, paper, anything, these are not just for only one set of faces. If I can provide you one of the most
it is known as sample bias, you are right, and you can say that people with different ethnicity are not very much considered while making the data set, and that results in drastic, you can say, performance drop. And I was once working on data. It was data set. It was for crop disease prediction, and when we were working on real data, the performance got really very bad because all the images that we were having were clicked in the laboratory. They were having white backgrounds, and when real data was put, it was you can say pretty much not similar to the data that was given for the training. So yeah, the lighting conditions and the quality, image quality, and the background and everything's really, you can say, affect the data. You may be getting good training loss and good accuracy by trying, but when it comes to real world, the table get changes and the whole picture is somewhat very different. That is a thing that is, you can say, very much ignored in data science. That how people only, count how many images you are having, how many rows and columns you are having, but how they are related to the real world and the quality of the data is very much ignored. So that's it. Any questions?
Not a question, but I really like what you're doing, Krishna. Good work.
Yeah, actually, we are doing it here on the paper with me.
Yeah. And I have to kind of give a more of an update. I'll do an update later this week on the New York celebration of women in computing stuff, which is related to bias and ethics in AI. But this is obviously this all ties together for that, and for people who don't know much about that, we're part with the other group. We're working on some AI ethics related stuff. So if you're interested in AI ethics and bias sort of things, just mention it in Slack, and we can talk more about it there. But this is the bias paper is definitely was one part of that, and I like where it's going. And listing out the biases is, it's something that we kind of all have to be familiar with going forward. So it's good.
Yeah.
So I have to present one more thing. It's regarding the conference that
Okay.
So it's not completed. It's just a super resolution. So, give an example, when we have data that is very much cropped and the pixels have to dilate and not much, guess that what the thing really is. We have into a technique and it's not a super resolution. Okay. So there are two types to perform the task of super resolution. One is bicubic interpolation, and other is with the help of CNN, Vadi, abstract the features. So I'll be explaining the bicubic interpolation. It's quite a, you can say, a math heavy topic. It's quite complex. So I'll just tell that if you want to know, interpolation is just like filling out the voids. And if we want to fill the voids by using the bicubic interpolation, we consider the things like what are the pixel gradients. For example, if I have a line here and I want to predict that if this pixel is missing out there, and I want to predict that how the pixel would look like, then I will what I will do, I consider what is the horizontal gradient. Gradient is, you can say, the change in the color. Like for example, in this text you can see there is a gradient. Not every color is same. We have some of it is pink here and it's yellow here, and it is something you can say intermediate here. So it is known as gradient. So here we are considering the gradient. For example, if this point has to be considered, then its horizontal and vertical gradient should be considered. So I'll not go much into mathematics and spare you people off. And there is another thing known as nearest neighbors. For example, if now I have to consider this block's gradient, this block's color, then I'll take value of K and interpolate it according to the closest 5 are of this color and the maximum color that is locating itself, I'll fill it with this color. That particular interpolation works better because it's not making the pixels into very discrete form. We still have some moving gradients here. It's not all 1 and 0 as the case with algorithm. So It's just an initial draft. I'll be, updating it in coming weeks for the Envision conference.
Okay. What is the? So this is basically upsampling data.
Yeah.
Okay. Why don't can you send me a link to the slides? I'd like to look them over.
Yeah, sure.
Yeah,
I'll share with you and Jesse.
All right. Yeah, that looks pretty good. I think we can find a home for that. I don't know. Like, we'll keep working on it, and we'll see.
Sure.
Thank you.
Yeah.
Any questions about that? I know Krishna's audio was a little off on it, but it's like, I think you got the idea. So yeah, I think that's if people have next week, if people have things that they're working on, they want to present, please feel free to present them, put them on the list. Let's see what we have in the chat. We have a lot of different things here that people have been posting. So Susan says axolotl was a model organism, so it's a salamander. Xenopus is it's a frog, yeah. And Akshay says my bio is weak. That's why I want to put some more tutorial information up on the onboarding guide, because I think people maybe need a refresher in biology. It helps. And we don't have any, for model organisms, you can look them up by their name that you see. If you see a name of a model organism, you'll be able to find out more. Like, there are a lot of references online, Wikipedia, just for a very basic level understanding of it. So this is, we have a lot of these model organisms. We've talked about this. Okay, this person does not exist. This is an example of a GAN where they generate people who don't exist, and they generate faces from, just existing human faces, but they're generating new faces and people who don't exist. So this is bias that Twitter exhibits. Twitter crops images to zoom into faces, and it's generally more biased towards white faces. So that's it's a design feature of some of this is kind of what we're talking about. And then NVIDIA uses super sampling on games. This is a link from Tharun. So this is a link to what they're using super sampling for. So some very good points in the chat. Thank you for contributing to the chat. Finally, I'd like to turn to some papers. I was hoping I had more time for this, and I'm not going to go through too much more, but I wanted to go over some points that I had prepared here. First of all, we continuing our discussion of cellular automata, I wanted to point people to this. There's been some interesting work on morphogenesis in Minecraft. And so this is something that's using three-dimensional cellular automata coupled to a neural network. So it's something they call neural cellular automata. And there's a new paper here on the archive on this topic. Sebastian Risi is one of the authors, and it talks about how they show that neural cellular automata can learn to grow complex 3D objects in Minecraft. So for those of you who are knowledgeable about what Minecraft is, it's a three-dimensional environment. It's a virtual environment where you can build things with blocks. So it's kind of like virtual Legos, and you can put them together and build like all sorts of things. And you can build avatars, and you can interact with each other in the environment. And what they're doing here is they're taking these blocks and they're using that block structure to generate 3D artifacts, and they're basically evolving them using this neural cellular automata, or they're forming them. I don't know if they actually use an evolutionary algorithm, but they're using this approach to build these artifacts. And they're actually also to show that you can build functional Minecraft machines that regenerate when you cut them in half. So this is like what you see with some worms. You see this with flatworms, this extreme ability to regenerate. But other organisms, if you cut them in half, they regenerate the rest of their body. So if you cut a single organism that exhibits this property in half, you can end up with two separate organisms. And so, this is an interesting advance. If you want to know more, you can read the paper on that. They also have I think I posted it in the Slack channel. There's a competition for open-ended evolution in Minecraft as well. So they're using Minecraft for all sorts of interesting things these days, and we should talk more about how to leverage that in future meetings. So let's see. I also had another question that was raised after last week's meeting by Dick Gordon, and we talked about some papers on the environment. And it kept using the word environment, and the papers kept using the word environment. And he had a question in the chat, and he kind of tried to clarify it after the meeting. The idea is that environment, we talk about it in development as being this thing that like an embryo exists in, or that organisms experience. But what is it? What is its relevance, and how do you define it? And it might seem like a very elementary question. And Dick is, he's done a lot of work in biology and development, so he knows. You'd think he would know what the answer is, but I don't think anyone knows what the answer is, actually. It's very unclear because people throw the term environment around, and it's like, well, what does it mean? And so a lot of times people will throw this term around and not really define it, or not really know what it does. And so we kind of targeted two different areas. The first is that environment is permissive. So environment allows for things to happen in the organism. So that may be some very general type of information, like changes in the environment, like from hot to cold, or stresses, so like, starvation versus food availability, or intensities. So you might have a temperature gradient, where you might get really hot, super hot, or super cold. And so those things can be transduced into the biological system. It might permit certain things to happen. It might retard certain things from happening. But it's this idea that it's permissive. It allows for things. And so, but the second is that environment is instructive. So this is different in the sense that environment actually has some specific information to give to the biological system. So that might include like ratios of different signaling molecules. Oftentimes there are signaling pathways in cells or in small microorganisms that allow for things called like quorum sensing or other types of collective behavior. And that also, involves patterns or codes that might be involved in the environment. Like, fluctuations in the environment are interpreted in there might be patterns in that sequence of events that trigger things in the biological system. So the environment is also instructive. And so this is sort of trying to work this problem out. And I don't know if people have things you want to add to this, so you can talk about it in the Slack. But I think it's an interesting sort of idea to think about. But, that's maybe something we can talk about later in future meetings. I'd also like to finally kind of get into some of the papers. I don't want to get too deeply into these papers. Jesse sent me this paper on nothing in evolution makes sense except in the light of parasites. And this is by a couple people who actually contribute to the artificial life literature, Susan Stepney and Pauline Hogeweg. And this is talking about sort of the role of parasites in evolution. And so they talk about, the emergence of parasites in evolving replicating systems appears to be inevitable. And so that means that parasites play this critical role in evolutionary systems. So this is something they talk about spatial pattern formation being a factor in, or parasites being a factor in spatial pattern formation. It also slows down replication sometimes. It involves things like coevolutionary arms races. And so there are a lot of things that parasites are involved in, or parasitism more generally, which is basically one organism or one entity benefiting off of another entity. So there are different types of interactive relationships. There's parasitism, there's mutualism, and there are different things like that. Parasitism is where you have one entity sort of living off of another entity without contributing anything. And so there's a lot of interesting stuff that this is actually, I think it may have some simulation work in it. I can't seem to get the thing to scroll down. There we go. So yeah, they have a lot of different, so they're using this stringable automata chemistry. So they're using an artificial chemistry to model some of this stuff. So it's really kind of an interesting approach to parasitism. And they look at the population dynamics. So you can see that they're mapping the population and number of species over time, and they're looking at they're modeling their hypotheses using this platform. And here's some, some screenshots of how they're evolving this artificial chemistry. So this is what it looks like. You're just evolving agents in this artificial chemistry, and you're putting together, you're getting these results. Or different chemical reactions. And so I'm not really an expert at artificial chemistry by far, but just to let you know that these things exist in the world and that you can do cool things with them. Then there's this other paper on modeling developmental sequences, this neurodevelopmental sequences. I only bring this up because it's something that's been out for a while, but I found it this week. They basically look at neural development in 18 mammalian species, and they look at like different heterochronic changes in brain evolution. So what that means is that they look at the timing of the development of the brains in all these different mammal species, and they look at what's going on. So they're looking at like how neurons form and connections grow and change over developmental time, and how connectivity is refined. And they have all these data that show that their timing changes and the onset of growth and the duration of growth and other things that actually lead to a lot of the changes you see in different species. And so this is a sort of a catalog of all these changes. It's a really complete paper. And I mean, this actually is something we might look into more. In the other group, we're doing some work on developmental AI, and this is something that might be very useful to that. But I think this is also useful for understanding development in terms of the brain. And then finally, we have a couple of papers here that a couple of things that are kind of coming out now that are really exciting. There's some wet lab work on an ex utero mouse embryogenesis model. So they actually have this ex utero mouse embryogenesis from pregastrulational late organogenesis. So this is a mouse model that is looking at formation of an embryo and then looking at differentiation within that embryo. And so this is done in a lab. This is done in cell culture. We've talked about what cell culture is. And they're looking at some of these changes that are happening in the embryo from the time when there's like this mass of cells that are starting to take shape to the formation of hind limbs. And so, you're actually getting not only tissues, but the formation of different parts of the phenotype. And so this is really interesting work. This is a very technical overview here. I don't know if I have the, this is actually something related to this. This is a News and Views article, so this is easier to understand. This is the first complete model of the human embryo. So in this case, they're not doing what they did with the mouse so much, but they're actually taking a human embryo and they're actually able to grow blastocyst in a dish. And so they're able to grow these human blastocyst-like structures. So this is the blastocyst, where it's a very simple version of what's going to happen in the formation of the human embryo. You're going to have this inner cell mass, and you have this trophectoderm layer. So you have a very early stage developmental structure here. And so studying early development in humans has been a challenge because it's hard to get, there are ethical considerations, but it's also hard to get these things to grow in a culture. And so, and to really study it like we would like to. That's why we use model organisms, in fact, because we can't really study these things in humans. One of the reasons why we use a lot of different model organisms. But now there are 2 papers in Nature that make key advances in this field, allowing us to actually reprogram cells from adult tissues to form these, or to use human embryonic stem cells and to form these blastocysts in a dish. And so basically they're going to get these blastoids, which are these sort of blastocyst aggregations or cells that behave like a human blastocyst. And so you're able to generate, what the advantage is here is that you can generate these without having to deal with a lot of the ethical constraints or with a lot of the the consequences of trying to get a human embryo to grow into a blastocyst outside of the womb. And so we've talked about different sorts of organoids before, organoids, gastroloids, and this is a blastoid. So this is really exciting work, and they just grow it in this chamber, and they let the cells aggregate and do this basic pattern formation, and they want to study what happens during this process. And so this is some more advances in this model. So I don't have the full papers are listed in the references of this. So often what they do is they have this overview paper and they cite the original papers in this, but it's a nice accessible way to understand it. And then this paper is a little bit deeper into the details of modeling embryogenesis in a dish, but they're doing this in mouse. And so these are a number of different models in mammalian systems, which are much different than our C. elegans and our other models that we're doing in our group. But they're very important to understand if you want to understand development. So we have some questions, and I'll take those before we go. So Susan says, I have some seashells. Should I take some 3D images of them? Yes, please, if you can. I'd love to see those. Mainak says, axolotls can regenerate. Yes, axolotls are a key model organism in regeneration studies. If you go to the regeneration literature, you'll find they use axolotls a lot. Ojwal says, environment is adaptive, which is true. So, we're trying to figure out what the role of environment is. But environment itself isn't adaptive. It drives adaptation in the organism. So, we're trying to piece this together. The language is very broad and general, and we want to kind of get this together. Can you post a link to the parasite paper? Yeah, I'll post it in the Slack channel. That would be a good thing for people to read, I think. Dick's here. Hello, Dick. Anyone willing to learn EndNote Basic? So if you want to get involved with EndNote, we want to put these papers in a bibliography of some type. Shruti says, was the issue resolved in the code? Dick says, axolotls develop at constant temperature, oxygen, and distilled water. What does environment do? And so this is the thing about the question about the environment. Funding was cut for human embryos decades ago. Yeah, they have not really funded that area. So, Shruti collected 100 papers for the paper I'm writing. Please be patient. And axolotls regenerate spinal cord and heart. Yes, axolotls are a very good model organism for looking at a lot of things in organogenesis as well. So thank you for meeting. I know we're probably at the end of our time here, and I just want to say, if we'll be going over some of this stuff on Slack. We should have some more discussions on Slack or via email. I send out the weekly email list, and so if you're interested in submitting a paper to present during the meeting, or if you have something to present, please bring it to the meeting or send it to me in advance, and we can do that. Any other questions before we go? Any other comments? Okay. Our ancestors look like axolotls. Why can't we regenerate limbs? I think that's a major question in the regenerative medicine community. I know that I've worked with people I worked with one person who did work on newts and other in other neural systems. And it's still hard for people to understand like why we can't regenerate. Like lizards and amphibians have some, and fishes as well, have some quite impressive abilities in terms of regeneration, but we don't have that. And there are a lot of theories as to why, but no one's really, I think, really uncovered the key factors and why that is. I mean, it's going to deserve like a presentation to get into the reasons why. I don't want to. So, Jesse, EndNote Basic is free. It's capacity for 50,000 records. So, that's... So if there are no more questions, thank you for meeting. Have a good week.